Thank you so much for watching this video. I'm Sepanta from the School of Dentistry, and I want to talk about part of my PhD project about microRNA 26A delivery by mesopore silica nanoparticles to mesenchymal stem cells in order to promote osteogenic differentiation. This graphical abstract is a schematic progress of the whole project. The most center circle, which is the MSN plus microRNA delivery system, is the beginning of the study. Following fabrication of core cone mesopore silicon nanoparticle and loading them with microRNA 26A, first I did characterize the nanoparticles, nanocomplexes, and then the cellular uptake and transfection efficiency were evaluated. And finally, the functionality of the whole platform was assessed. Now I just want to talk about a little bit about the background of this research. microRNA therapeutic application in bone regenerative medicine significantly raised recently. And the main advantages of microRNA-mediated tissue regeneration are a more sustained expression and prolonged regulatory activity. Actually, cytoplasmic half-life of many microRNAs spans longer than 24 hours to a couple of days in comparison to maximum 5 hours for plasmid DNA and 15 to a couple of hours for growth factors. And there's no immune reaction for microRNAs. So that's why microRNAs can be used for many regenerative purposes in dentistry. This slide is showing various microRNAs that can be involved in osteogenesis, osteoclastogenesis, and angiogenesis, which are the main components of bone regeneration. I mainly focus on microRNA that coupling angiogenesis and osteogenesis because, as you know, one of the major clinical issues for bone regeneration is neovascularization and angiogenesis. That's why we chose microRNA 26A, which is able to activate VEGF and WNT pathways at the same time. Although microRNA can be functional at low dosage and featuring longer half-life compared to growth factors and plasmid DNA, the delivery of plain microRNA has represented a limited success. We need an efficient and safe delivery system. For this purpose, we are using large pore size mesopore silica nanoparticles or core cone mesopore silica nanoparticles. These biocompatible nanoparticles are highly flexible in terms of surface modification and customization, which enable us to alter their structure according to the specific requirements. I don't want to go into the details about the methods because of lack of time and also the scope of this presentation. I just briefly explain what we have done during this research. First of all, we fabricate core cone mesopore silica nanoparticles, CCM, MSNCCs, and then coated them with PEI, polyethylene amine, and to increase the zeta potential and absorb nucleic acids, then characterize the nanoparticles with SEM and TEM analysis. The biocompatibility were assessed with MTT assay, and then loading capacity evaluated by nanodrop and transfection efficiency were evaluated by laser scanning, confocal microscopy, and also flow cytometry analysis. And finally, functionality assessment evaluated by osteogenic gene expression, RT-PCR, ALP activity assay, and the extracellular matrix deposition and mineralization were assessed by Picker-Series red and alizarin red staining. These are the results of characterization of nanoparticles before and after PI cutting. TEM and SEM analysis and also nitrogen absorption analysis did not show any significant change after coating. Also in this slide, dynamic light scattering analysis confirmed the previous results and did not show any significant change after coating. And as we discussed before, zeta potential increased following the coating with PI, and it was nearly 36 millivolt higher than MSNCC. Panel D is showing that loading capacity of MSNCC-PI is high, and the higher the concentration of nanoparticles, the more the microRNA loaded. The percentage of viable cells were evaluated at 1, 3, and 7 days after exposure to nanoparticles, and based on these results, besides the loading capacity findings, 10, 20, and 40 microgram of MSN were chosen for further evaluations. As you can see here, the confocal microscopy images, small green dots are fluorescent labeled microRNA, which has been taken by cells and currently are in the cytoplasm, and big blue dots are nuclei which were stained by DAPI. In accordance to the confocal images, phagocytometry also showed efficient transfection rate between 61.1 to 86.5% for 10 to 40 microgram of MSN respectively. And also mean fluorescent intensity increased in a dose-dependent manner. Based on these findings, 40 microgram of MSN considered to be assessed in the following steps because of its high transfection efficiency and low cytotoxicity. Now, with having an efficient delivery system, we focus on the impact of microRNA 26A in terms of osteogenic differentiation of bone marrow mesenchymal stem cells. In this regard, we use microRNA 26A mimic as the experimental group and microRNA 26 inhibitory, negative control, and microRNA and untreated cells as the control group. And we evaluated ALP activity. You can see it in panel A and 7 osteogenic gene markers from panel B to panel H. All of them except GSK3 beta significantly increased by microRNA 26 mimic induction compared to control. GSK3 beta, or glycogen synthesis kinase 3 beta, is encoding an enzyme which inhibits beta-catenin pathway. This pathway is essential for osteogenesis. So inhibiting this, the expression of this enzyme can increase and enhance bone regeneration. In this slide, in accordance to previous results, collagen secreted by BMSCs, bone marrow mesenchymal stem cells, after 21 days of incubation showed significant improvement. And you can see microRNA 26A mimic group in panel A, and other panels actually are just control groups. Also, in addition to that, a quantification of the staining showed significant increase in the release of, in the relative level of collagen in microRNA 26A group compared to other groups to controls. And finally, the alizarin staining also confirmed previous results. And as you can see in panel A, there are many mineralized calcium nodules that form by microRNA induction. Panel E also shows the comparison, the quantitatively comparison of the staining results. In conclusion, our results actually support the concept that microRNA 26A mimic can be effectively delivered into mesenchymal stem cells by using MSNCC-PI as a vector and to promote osteogenic differentiation in these cells. There are many potential applications of this delivery system, this platform, such as bone regeneration, specifically in large bone defects, bone-related diseases such as osteoporosis and hard tissue formation in regenerative dentistry. I'm currently focusing on the responsiveness and incorporating this platform into its scaffolds. Thank you so much for watching this video again, and these are my contact details. I will be more than happy to answer your potential questions and a discussion for any kind of collaboration. Thank you.